Nagaraj sir?
Sir, we will start.
Yes, sir. Yes, sir.
Sir is joined, no? Bharat sir.
Just confirm with him, sir.
Yes, sir.
Bharat?
Yes, sir.
Okay, sir.
Okay,
Good
sir.
morning.
Good morning, sir.
Uh,
I'm the first person to join in the meeting, sir. By
Oh.
9:30, I joined.
Thank you, sir.
Thank you,
Thank you,
sir.
sir. Thank you. Thank
Sir. Thank you, sir.
Sir, I'm starting. Gandhi sir.
you. Go ahead, sir. Go ahead.
Yes. Yeah. Good morning to all. Welcome to the last day of online training program on integrating network pharmacology with traditional and modern system of medicine, bridging multi-system therapeutics for holistic healthcare. So last 5 days we had a wonderful and knowledgeable sessions. So I hope you all are got the knowledge from that session. So today also we have wonderful knowledgeable session by Dr. S. Bharatkumar Reddy sir. So all I am requesting, please all of you utilize this session and ask the some questions to the sir and get the knowledge from this session also. So without any further delay, so we will start the session. So now I welcome Dr. Mohan Gandhi sir, Professor, Department of Pharmaceutical Quality Assurance, to introduce the today resource person.
Thank you, sir. I'm delighted to introduce our esteemed guest speaker, Dr. S. Bharat Kumar Reddy, who completed his Master of Pharmacy in Pharmacology at JSS College of Pharmacy, Ooty, Tamil Nadu, where he graduated with distinction. Dr. Reddy further pursued his Doctor of Philosophy, PhD, at JSS Academy of Higher Education and Research, Mysuru, Karnataka, supported by the prestigious ICMR Senior Research Fellowship designation. Dr. Reddy's remarkable contributions to the field of pharmacology have been recognized through various accolades and achievements. He has received the Best Scholar Award from the National Educational Brilliance Award, the Young Researcher Award, and the Research Excellence Award from the Institute of Scholars. Additionally, Dr. Reddy has been acknowledged for his research with several travel grants, Best Poster Award at national and international conferences, and academic excellence during his graduation program. With a strong publication record, Dr. Reddy has significantly contributed to the field with his research work accumulating a notable cumulative impact factor of 110.5. His innovative research has led to the filing and granting of several patents, showcasing his commitment to advancing pharmaceutical research and innovations. Dr. Dr. Reddy's expertise in investigating the effects of drugs and natural compounds, coupled with his extensive experience in pharmacological and toxicological assessments, molecular and cellular studies, and computational approaches, positions him as a distinguished expert in the field. With this brief introduction, now I would like to invite today's resource person, Dr. S. Bharat Kumar Reddy, sir. Sir, the forum is to you. Thank you. Just a minute, sir. I'll host you. Now you please check, sir.
Sir, can you confirm whether my screen is visible?
Yes, sir. It is visible, sir. Please
Okay.
maximize it.
Okay. So I'm audible too. Okay, I need to maximize it, right?
Yes, sir.
So,
Perfect, sir.
okay, done. Good morning, everyone. I would like to thank our professor for the kind introduction. I also appreciate all of you joining us today for the final session of our 1-week training program. So before we dive into today's topic, I would like to make one thing very clear that within the next hour, I won't be presenting any revolutionary or overly complex concepts, but instead we will explore familiar ideas that too from a fresh perspective. Means you will likely to hear few terms that you were already familiar within this last 1 week, but I hope to provide new insights and connections. I will try my best. So try to be interactive. If you are having any doubts in between, you can feel free to interrupt me at any time. So let's get started. So network pharmacology, I think from last 1 week, all of you are hearing about this, a commonly known definition. Even today, you are going to hear the same, but in a little different way, and that in a little deeper manner. The first, network pharmacology. So it's a new and emerging field that specially focuses on understanding diseases and drug actions, especially from a perspective of systems biology rather than focusing on OTAT. So here OTAT refers to one target at a time. This network pharmacology focuses on MTAT, that is nothing but multiple targets at a single time. So when I'm saying this particular definition, network pharmacology, I have used a term that is perspective of system biology. So what does this system biology refers to? Can anyone think it of? So system biology means it's like looking a biological system whole rather than a rather than focusing on an individual component. So when I'm saying it's as a looking as a whole biological system, it's the system refers to the humans, means it is us. So what it is going to be do and what sort of things that we are going to look over here one by one we will see. The first thing, so this approach, whatever the approach that this system biology is going to be used, it is going to be helpful for us in order to assess this highly interconnected biological process, and in order to understand this typical multifaceted pathophysiological diseases, as these diseases arises from a different set of or a different type of pathophysiologists together create a particular disease. So this systemic approach will help us to assess all these aspects, and because of that, this is also going to help us to map how drugs are going to interact with these multiple targets in the body. So now we had seen what is the system biology. So now we will see how this system biology is going to integrate its particular data with the network pharmacology. Generally, this network pharmacology relies on a diverse set of biological data. So why this network pharmacology is relying on this diverse set of biological data? Because in order to create a comprehensive view how these drugs and diseases are going to interact, this is going to depends on a diverse set of data. So when I'm saying this term diverse set of data, you just think, so what is this diverse set and how this network pharmacology is going to get this diverse set of data? So no doubt this diverse set of data we are going to get because of a various high throughput technologies that has been captured from different aspects of biology. So when I'm saying different aspects of biology, what and all going to be considered over here is like the first thing is genomics, second is proteomics, followed by transcriptomics and metabolomics. So now what is the genomic refers means one by one we will see what each term refers to and how this is going to be integrated in the network pharmacology. We will see. The first thing is genomics. So genomics, it's a self-explanatory word, means it says about a gene. So it's a study of complete or entire genomic sequence of a particular organ. So when we are saying study of entire genomic sequence, here we are going to study the sequence length, structure, and functions of all the genes. So how it is going to be helpful in the network pharmacology? This is the first question. So when we see this genomic data helps us to identify variations or genetic variations or mutations. So most commonly when we say mutation, the first disease that comes to your mind is cancer. For example, if you say mutation in a particular gene may cause cancer. So because of this, there will be alteration in a signaling pathways. So in order to getting an overview or a knowledge related to this genomic data will help us to focus on that particular mutated gene such that an efficient drug can be designed to target that particular pathway. So the next term, that is proteomics. So same like genomics, this is also a self-explanatory word. So when we say this proteomics, this is a large scale study of proteins, particularly their function, structure, and interactions. So why we need to study about this proteins? So once if you go back to the structural unit of this receptors, so they are amino acids together form peptides, peptides together form polypeptides, polypeptides together form proteins, and in few cases this proteins itself acts as a target, and in few cases these proteins together form receptor. So now if you see, these proteins are going to be a molecular machines that carry out most of the functions in the body, and they also acts as a key player in the pathways that are going to be contributing to a particular disease. So for example, if you see in terms of inflammation, either in case of diabetic wound or autoimmune diseases, if you see, interleukin 1, 6, 10 or TNF alpha, all these were the proteins. But because of this proteomic data, we have got a clear idea that these were the inflammatory mediators responsible for the cause of this particular immune responses. So that is how it became a target for us to design a suitable drugs for this particular protein. The next term is transcriptomics. So in terms of genomics, we had seen a data related to the genes, that is nothing but DNA. Now in terms of transcriptomics, this is the one which, this is the one where we are going to study about RNA molecules. When I'm saying about RNA molecules, here all types of RNAs are being involved, even messenger RNA too will be involved. So as this mRNA is the direct product of gene expression, that is why this transcriptomics data will help us to provide the insights into which genes are going to be active in a specific cell or tissue. So now all these terms are the last but not least, that is nothing but metabolomics. In terms of metabolomics, it is going to be a large scale of study of metabolites. So these are nothing but a byproducts of metabolism. This also we are going to study. So how these all four terminologies, it is going to be incorporated in the network pharmacology. Thing is like the network pharmacology, all these terminologies we had seen will help us to identify a particular gene, protein, or an RNA or a metabolite that is being involved in a particular disease expression. So because of this particular identification, means here we are going to identify whether the particular gene, protein or RNA is overexpressed or underexpressed. Because of this identification, we will get a clear picture to target which particular part. Means either by if you get that particular idea, we can either design a drug that is having an ability to silence these harmful genes, or we can also design a drug in order to enhance the beneficial one which is already suppressed in the disease condition. This is how the system biology is going to be helpful. So next, coming to this holistic approach, means all this till now I am discussing about this network pharmacology definition only. So we have used a few terms, that is system biology integration and holistic approach. So now we have completed the system biology integration. So what does it mean? So now holistic approach in understanding a disease and therapeutic intervention. So when we say this term holistic approach over here, what to say, this network pharmacology is integrating the data from different aspects. So that is nothing but already we have discussed in terms of system biology. It is integrating the data from genomics, proteomics, transcriptomics. In order to understand these networks, there need to be a separate set of approaches we need to require. So that is where this holistic approach came into existence. You can just take an example of this cancer. In terms of cancer, we can see several genes and proteins or pathways. All these are going to play like an interplay used to happen between these genes, proteins, and pathways. Together, they used to promote a tumor growth or metastasis, or they also helps in creating a resistance to the treatment. As we are going to use this particular system biology integration in terms of network pharmacology, it is going to be very easy for us to map these molecular targets or molecular networks, and we can also identify the targets for a particular drug rather than focusing on a single mutation, that is nothing but OTAT, which we have already discussed. Means a networking can be efficiently performed over here. So as per the title, the first thing we have completed is like network pharmacology. Next, we will move on to the next aspect of our title, that is nothing but integrative medicine. So what does this integrative medicine refers to? So even this is a known thing for you, but in a different term they are saying. So when we say integrative medicine, it's a holistic approach to the healthcare system where they are combining this conventional uh, therapy with that of complementary medicine. So please focus here when I'm saying conventional therapy. So this conventional therapy is confined to the Western approach. For example, even a layman knows if a person got affected with a cancer, so immediately they will say that the person need to be treated with some sort of chemotherapeutic agents. So if not, it is followed by a radiotherapy, not followed by a surgery. Means conventionally as a standard care, these are being used from a long time in terms of Western medicine. Now, taking that standard or conventional Western medicine, that we are combining with that of complementary medicine. So when we say complementary medicine, it is nothing but alternative of allopathy. It's like Ayurveda, traditional Chinese medicines, homeopathy, Unani, chiropractic, and so on. Means combining these two is nothing but integrative medicine. So the major purpose of this integrative medicine is to prioritize the whole person body rather than treating a single thing. So coming to this principles, key principles of this particular integrative medicine, the key principles are stated over here, that is like a patient-centric care, holistic approaches, evidence-based treatments, and collaborations. So when we say patient-centric care, we will recall the few known examples. I think all of us might be aware about this drug, that is isoniazid. It's a well-known anti-tubercular drug. So when we think about this anti-tubercular drug, and if you compare the different set of populations between Asian and Western people, Asians are going to be the fast accelerators. The same thing if you see in terms of Western population, they are going to be slow accelerators. So why this is happening? This is because of a phase 2 metabolism or phase 2 reaction that is being means a acetylation process in order to acetylate this particular drug. So this acetylation, why it is fast and slow? Because of overexpression of a gene known as N-acetyltransferase. So in terms of what to say, Asian population, this N-acetyltransferase gene is being overexpressed compared to the Western people. So when I'm saying here N-acetyltransferase gene, so here comes this patient-centric care. Because of this identification of this particular overexpressed or underexpressed gene, accordingly, a tailored medicine can be designed for that particular set of population. In the same way, we can still narrow down the procedure to each of the patient. So based upon their special data related to the genome sequence or transcriptome sequence and so on, we can incorporate or we can choose an alternative therapy such that the patient outcome can be made very efficiently. So we will see few of the case studies that are related to this network pharmacology as well as integrative medicine. The first case study that I'm going to discuss today is about diabetic wound. I think you might be aware, diabetic wound is one of the most devastating complication of diabetes mellitus. Devastating complication of diabetes mellitus. So what is happening over here means it generally this diabetic wound healing means the wound healing in a diabetic patients generally gets delayed because of the interplay between triopathy condition. So when I'm saying a triopathy, it's a combination of a neuropathy, immunopathy, as well as vasculopathy. So as the pathophysiology means as the process is being interplayed between these three, the wound is getting delayed. So now how this integrative medicine approach is going to be helpful in terms of promoting this diabetic wound healing faster. So we will see an example, I think, and the current trend in terms of formulation is nanoparticles. So nanoparticle is one of a conventional therapy we can say in terms of westernized or allopathic system. So what to say, selecting a polymer to the nanoparticles which are having a role in terms of wound healing is one such approach. Along with this conventional, we can choose or add an existing traditional or herbs. So the best example or approach that has been performed by one of our colleagues is curcumin nanoparticles. So the scientific name, or I think we might be aware, it's a curcumin longa. So curcumin, if you see, it's a well-known traditional medicine which is having anti-inflammatory properties. Even we can recall our parents or our grandparents immediately when we have been injured with some sort of wound, so they will sterilize over there and immediately they will start applying this curcumin, so which is having a anti-inflammatory property. So now how this network pharmacology has helped and how it is being correlated, all of a sudden from a definition it has been shifted to a case study. So let's see how it is going to be connected. The first thing is like what to say, already in the conventional dosage form nanoparticles are being used. So in terms of integrative medicine, curcumin is one of a natural ayurvedic one which is having a property of anti-inflammation. As in terms of diabetic wound healing, the wound is being struck in the chronic inflammatory phase. So choosing a compound or a natural herb which is having an anti-inflammatory property and incorporating these in a suitable dosage form and applying it topically is one thing. The second thing is like addressing the lifestyle changes. So when I'm saying addressing the lifestyle changes, in the sense it is through dietary interventions or complementary practices such as acupunctures in order to promote circulation. So now you correlate the example here very clearly, because see, in a traditional treatment we are giving insulin side by, because if the diabetes is not controlled, wound healing cannot be promoted. So with the help of traditional medicine, side by we are giving insulin to control the chronic diabetic condition. Simultaneously, we are also giving the alternative therapy that is nothing but curcumin loaded nanoparticles for wound healing. So this comes here. It is an integrative medicine approach. So this has been identified with the help of this particular network pharmacology. So in the same way, even we can see one more example that is related to uh, cancer. So prostate cancer, just okay. So it's related to ovarian cancer. So here also, how this network pharmacology has helped in treating this particular disease effectively. I think in terms of chemotherapy, we might be aware about this drug that is known as cisplatin. So it's a well-known drug that we are going to be commonly used for treating cancer. So in network pharmacology supports the integration. So how this means what is this case study completely deals about? So this network pharmacology support helps in integrating and analyzing the multiple components and targets that are having an ability to interact with this therapeutic efficacy, optimize the therapeutic efficacy. Actually, one of it's data from a published paper where the authors have investigated this curcumin longa. Mm, yes. So, the authors have investigated the use of curcumin longa and they have find out with which drug we can combine it and whether there is any chances or feasibility to enhance the process of further metastasis or angiogenesis in the cancer cell. And in this research, they have efficiently found out that initially they have they have used this network pharmacology and they have revealed this curcumin means already it is known for its anti-inflammatory property. So both together they have merged and identified that curcumin is having an ability to target proteins that is nothing but VEGF, vascular endothelial growth factor, which is responsible for angiogenesis, and MMPs, which are responsible for prolonged inflammation. So curcumin is having an ability to inhibit both of these VEGF and MMPs. So now combining the cisplatin along with this curcumin, once again it falls under an integrative approach means a combining of conventional treatment that is nothing but cisplatin along with complementary medicine that is nothing but use of curcumin to so finally the outcome is like the combined treatment. The authors have demonstrated that there is a drastic shrinkage in the tumor size and even they had seen there is an increase in the survival rates compared to the use of single cisplatin alone. So in the same way, one more example in terms of antibacterials. So why I am discussing this case studies in the sense in order to sensitize you that how this network pharmacology approach is going to be helpful to us by incorporating different types of complementary medicines and how we can progress further for the well-being of patients. So the second thing is like bacterial infections. So I think we might be aware that the world is fastly progressing or it is fastly entering into an era of antibacterial resistance. And in few areas already the population has entered in this an era of antibacterial resistance. I think all of us might be aware about the deaths that has been caused by this COVID-19, right? But if you see the statistics, what to say, there is thrice higher deaths that has occurred in 2019 just because of bacterial infections means the deaths compared to the COVID-19. So this bacterial infections has become one of a major challenge to us. Now, what the authors have reported is like they have taken the use of network pharmacology where they have chosen a plant that is known as Baikal skullcap. Baikal skull cap. With the help of network pharmacology, they have identified two constituents. One is bicalin and another one is bicalene. So these two chemical constituents they have identified, and from the network pharmacology they tried to dig out that combining these two systemically identified compounds are having any sort of synergistic effect with the conventional antibiotics or not, and they have successfully established this, and the final outcome is these two bicalin and bicalene, these two chemical constituents, when they have been conjugated or administered along with the conventional antibiotics, there is increased in the inhibition of cell wall synthesis. Even there is increased in the sensitization of the bacterial death to the conventional antibiotics. Once again, this approach has been identified with the help of network pharmacology and integrative medicine. So now, when we are saying about all this case study, so I think this also you might be aware, and you have been studying about the stages that have been involved in the network pharmacology from last 1 week. The same thing over here, but what difference that we are going to see over here is different types of softwares that are being freely available, which will be helpful for you to perform one or the other way, means like your study can be efficiently performed. So that we are going to see in next coming slides. The first, the stages in network pharmacology, the first thing is identifying and collecting the biological data. One thing we need to make very clear, that framing of research hypothesis. Most of the time, now where the researchers are going to do mistake is lack of literature search. So initially they will start searching one or two sites, that mostly Google Scholar, and they will come back stating that there is no literature exists and I am the first person to work on this. So don't get too excited when the literature data is not exist. That doesn't mean you are the one or you are the first person to work on that, and it doesn't shows that it's an efficient research. When we are saying we are performing some sort of research, it should be properly backed with the supporting data. So that might be in terms of disease oriented, or that data from a complementary medicine, or data from similarity search. Somehow it need to be connected. Based upon that, we need to identify the biological data. So later on, it is followed by the disease as well as drug interaction networking. Next, protein-protein interaction networking, followed by biological mappings, drug target analyzing, topologies, drug repurposing analysis, and vice versa. And finally, at the end, it ends with molecular docking. So this is how the route map used to be there in all these stages. Yes. So here I am going to debit few computational tools or servers that is going to be helpful for your regular research works. The first stage is identification of active compounds or phytochemicals. I have provided you few commonly used tools or servers for the identification of this active compounds or phytochemicals. The most commonly used is IMPACT 2.0 database. Apart from that, KNAP SAC, this is another database. So where it provides a clear data about the plant species and their metabolites along with their bioactive compounds. The another thing is Duke's phytochemical and ethnobotanical database. So this particular tool or server used to provide a data on phytochemicals and their sources. So followed by TCMS and PhytoHub. So all these tools or servers, they are going to provide, or they are going to help us to identify the active compounds or phytochemicals that are being present in the selected plant. Next. So these were the tools that are going to be helpful for identifying the canonical smiles, molecular weight, and other chemical information. So the first well-known thing is like PubChem, followed by ChemSpider, Chembel, Zinc database, and drug banks. Their respective links has been provided over here, and even this PPT will be shared with the um, uh, Sandeep University people, so you can take help of this. So the purpose of the PubChem, I think we are all aware that it is going to provide a data on canonical smiles, molecular weights, chemical informations and all. Even the ChemSpider, Chembel, Zinc database, most of the time all these softwares provide similar type of data, and all these tools or servers are completely free, because most of the time getting a licensed version will be difficult because of some of the other financial constraints. So that is why the most commonly used freewares has been incorporated over here for the benefit of you. The next tools that are going to be helpful in this particular network pharmacology research is predicting the drug likeness properties. So in order to see whether the particular chemical constituent is having an ability to act as a drug or not, for which these were the commonly used tools or servers. So one is MoleSoft drug likeness calculators, Swiss ADME, Chemaxon, ADMET. More or less, all these tools predict the likeliness by assessing these log p values, their hydrogen acceptor capacity, donating capacity, topological bond surface area, their synthetic accessibility. Though sometimes what happens is like in terms of in silico techniques like network pharmacology or molecular docking, we will get a blockbuster compound which shows a very good docking score or very good p-values and all. But if you see the synthetic feasibility, it is very difficult for us to control few of the aspects for the synthesis of that particular compound. So here in terms of drug likeliness, even synthetic accessibility is the one thing that is being monitored. And even here, most of them were free. And in terms of Kenaxon, only the basic version is going to be free. If you want to use the complete details or complete full-fledged software, we need to go for licensing, and the same will implies to this AdMed lab also. Next, prediction of bioavailability. So once again, the Swiss ADME is going to be helpful, which is also shown its role in terms of assessing the drug likeness properties, and ADMED lab, PKCSM, pre-ADMED, FRP, and all. So where all these servers, once again, Swiss ADMA is most commonly used as this is going to be a freeware, and in terms of ADMED, again, its a basic version is going to be free. Whereas in the for the use of full-fledged software, we need to go for purchasing a license. Next, tools for predicting the toxicity of the compound. Protox 2, again ADMID lab, PKCSM, Vega QSR. So all these servers, once again, we can use for predicting the compound's toxicity. Means these are all the preliminary screening steps before entering into the full-fledged process of network pharmacology. This is how we are going to filter the compounds. Next, target predictions. For target predictions, we are going to use mostly Stitch software, Campbell, Swiss target prediction, Farm Mapper, SuperPreds and all. And all these tool prediction softwares which have been depicted over here are completely free. So once this that is done, next we will go for gene targets and their predictions. So again, for this, the information related to the genes, we are going to mostly retrieve from the gene card database, where we will get mostly the information related to all of the human genes, which has been what to say, identified till date. So apart from gene cards, there are few other databases, that is string, target scan, disgent and all. So where all these tools are servers, once again, they are free. So next, once this identification is done, then in order to assess uh, out of selected compounds, how many number of constituents are having an ability to interact with the receptor, for which there will be a plot diagram that is known as Venn plot. So for drawing this particular Venn plot, the commonly used tools or servers are going to be Venni and BioVenn. Again, once again, these two are free. So next, followed by protein-protein interaction. So for this protein-protein interaction, the commonly used server is going to be string. Apart from this string, there are few other softwares which are free, that is BioGrid, InAct, Reactome and all. So where these tools plays a major role in identifying the interaction between proteins. So next, once that interaction is being done, next for construction of this particular network between these diseases, drugs, genes and all, uh, these tools or servers can be used. Cytoscape. So whatever it has been depicted, all these were the parts of cytoscape. However, they have been segregated for each of their individual aspects. All this comes under a cytoscape software only. Once again, these were also free. So the next thing is like gene ontology enrichment analysis. Again, for this, we are going to use this particular enrichment tools, Metascape, Shiny, Enrich, and David. So where they will take they will start analyzing the list of genes that are being available in this KIC and other databases, and that is how they will start providing this enrichment analysis. And once this all network pharmacology is done, finally it is going to end up with this molecular docking. So in molecular docking, there are several free ways available, but Schrodinger is one of the licensed tool that is also being used several times in terms of papers. Apart from this Schrodinger, this Autodock, TorDock, and one-click dock, and several docking tools are there. This can be used for performing the molecular docking analysis. So now, so what are all the major challenges associated with this network pharmacology? As mostly in the starting itself, we had seen it's a new and emerging field. Slowly, the interest is being shifting towards this. So then, what are all the challenges? Because we need to look both sides, pros and cons. So the challenges will be in this way. The first thing is like data quality and availability. So initially, we had seen we are going to get chemical constituents from different websites, right? So once if the data set, whatever we are going to get is incomplete, the end conclusion that we are going to get will be mostly not accurate. Rather we can say that correlating with the clinical outcomes will become very difficult. Second thing is like complexity that is being associated with the biological systems. And we know that human system is a completely intricate in nature and it's very complex. So it will be little bit difficult for us in the initial stages. Means as of now, it will be very difficult for us to analyze and interpret this vast data that we are going to have. Next thing is like clinical outcomes, means integrating the same with this clinical practices. So whatever the thing that we are discussing, the network pharmacology and all, this is still the Cinderella, we can say, means it is still in its infancy, means it is not completely elucidated yet. So in this scenario, integrating this with the regular clinical practices once again will pose a significant challenge in the current stage as of now. And just a second, I just want to, I think we have discussed about few of the tools and we had seen sometimes means I have depicted that for each one of the stage, different sort of databases are going, like we are going to use. But however, I will try to show one sort of database, that is impact. So earlier there are only few headings that are being provided in this impact. So however, it is been little bit what to say added to this particular database. So this one impact database can provide most of the data that we require for this particular network pharmacology. So we will just explore over here. Hope my screen is visible to everyone. So this is an impact database. So this is how our home looks like. So initially in the impact database, so I'm typing this Tulasi. So these were all the plant parts that we are going to get, and chemical constituents along with their phytochemical names and their references from where they have been taken. So see, I have selected one of the constituents. So what and all data that we are going to get is like the first thing, summary. Apart from that, they are also providing their SMILES notations, physiochemical properties, drug likeness properties, their toxicology, admit properties, descriptors, all these things they are providing. So, but in now what they have done is like recently they have made some sort of advancements over here. See, initially I have typed Tulasi. So now I am giving avocado scientific name. Here if you see, there is one more new add-on that has been given over here. This is predicted human target proteins. This is lagging in terms of Tulasi. So all these data, this predicted human target protein is available for the few plant constituents as of now. So what sort of information that we are going to get in this human predicted human target proteins is the same data that we are going to get after completion of this network pharmacology. Means this impact database, they had a tie-up with this speech database. So directly they are providing the scores, interaction scores for few of the chemical moieties that have been identified. So this is one add-on that has been done recently. And that too, as of now, it is only for a few plant compounds. It is not available for all the plants. So as we have been evident that in terms of Tulasi, this option has been lagging. So now the next thing is like we have discussed about this molecular docking at the end. All right. So a little focus will be done on this particular molecular docking also. Hope I think yesterday few of you has raised some sort of concerns related to this. So generally this molecular docking, if you say, it's an approach from side of bioinformatics, chemistry, pharmacology, all together combines form this molecular docking. So what exactly we are going to see and why this molecular docking is essential at the end of network pharmacology. So when we say molecular docking, already in the initial slides I have discussed the what is that? The functional unit of this particular protein is amino acid. So our body is completely means whatever the targets that we are having, it is completely made up of amino acid. So when any sort of exogenous or endogenous compounds or drugs or ligands, if it want to show some sort of activity, they need to go and bind with that of a protein or receptor, right? So in a grass level, if you see, these are all composed with that of amino acids. So now even here in terms of network pharmacology, once after identifying these particular targets, we are going to we are going to see whether the identified targets is, means our drug is having an ability to interact with that identified targets or not, we are going to assess in terms of molecular docking. So in molecular docking, two things we need to remember. So the first thing is like ligand. So ligand can be of any small molecule or any protein or any enzyme which is having an ability to interact with the receptor or a protein and alter the state of that particular receptor either into active or inactive, thereby the following secondary messenger pathways will take on. All right. So the next thing is like protein. So protein, when we say, whoever the researchers around the world who are working on this particular proteins, So once the isolation is done, they will start dumping all these isolated proteins in a different sort of databases. That is nothing but PDB. PDB refers to Protein Data Bank. So here, whatever the site that I have opened is RCSB PDB. RCSB refers to Research Collaboratory Structural Bioinformatics. Apart from RCSB PDB, there are different sites also that have been available, but this is the one which is most commonly used for molecular docking and based upon the existing literature. So how this site is going to be helpful to us? Assume that we are the one, or the researchers are working on some sort of inflammatory mediators. So like MMPs. So this is how the homepage look like. So in the search term, we can type MMP. So there are different types of MMPs once again. So there is 1, 2, 3. My choice of interest is going to be 9. So I'm selecting MMP 9. So immediately when I select MMP9 and hit the search button, you can see there are different set of data that is being popping out over here. So what is this 1ITV? What is 5TH6, 5TH9, 1GKC? So this is known as PDB ID, nothing but Protein Data Bank ID. So how immediately after birth we are being named by our parents. In the same way, once the researcher isolate a particular protein or enzyme or a particular receptor, they will give some sort of names and they will be deposited in this data bank. So now I think you may get a doubt that for MMP9 there are this many results. So how we need to select and why we need to select a particular PDB ID. So there are several set of guidelines or criterias to select a PDB. So the first criteria is going to be the expression system. So assume that I have selected the first one I am selecting. So when I am saying MMP, so please be more specific over here. Based upon the target and the place where we are going to work, we need to select the organism. For example, when I am discussing about FTSZ, so FTSZ is a target that is specific to the bacteria. So in that case, if you are looking for an organism that is human, it is a blunder mistake. So it will be existing in the bacteria and my target of interest is antibacterial activity. That is why I need to look for FTS enzyme where the organism will be bacteria, not Homo sapien. But same thing in case of MMPs or any other diseases which we are working mostly in the human related aspects. At that time, we need to be more specific that the organism should be Homo sapien. The next thing is like expression system. So expression system can be anything. There is no compulsion for this expression system. Mostly E. coli will be the expression system in most of the PDB IDs. Next, we need to focus on mutations. We need to be more clear that until and unless we are working on a mutated receptor, we should not select the mutations. Means here you see mutations are 0, means no. So mostly mutations should not be there. That's what I have stated. Until and unless we are not working on a mutated receptor, we need to take care that mutations in a chosen PDB ID should be no. If not, there are chances of getting false results. So once this is done, so the next thing we need to look is like method of extraction. Already I have stated the researchers will identify some sort of proteins and again they will dump in a database. So what is this method then? So when I'm saying the researchers will identify the proteins or they will try to isolate the proteins, there are different set of methods. The commonly used methods for this isolation of these protein structures are X-ray diffraction. Second thing is like electron microscopy and NMR. These three methods are going to be commonly used for the isolation of a particular protein. Out of these three also, the most preferred structure is going to be X-ray diffraction. So why X-ray diffraction? Why not other two? For the X-ray diffraction structures, we will have resolution. In numerical, if you say lower the number, greater the resolution. Don't get confused over here. For example, here it is 1.95 angstrom. Same thing, it is 0.95 angstroms. Means the number, in terms of number, it is low, but resolution is higher. So lower the number, greater the stability it means. So whenever we are choosing a sort of receptor, we need to see that the structure should be more stable. So that is why we will go for least resolution. The other thing that we need to remember is like co-crystal. So when I'm saying a term co-crystal, this is a chemical moiety that is been already associated in the structure that is been isolated by the researchers. For example, here if you see, this is a completely structure of a particular protein. In between we can see there are few metal ions or few structures. So why this co-crystal is being required? See, slowly I'm going down. So here we can get a clear data about this particular macromolecule. Whatever I have chosen, the molecule is MMP9. Okay, it is showing over here. Now, if you go to the in-depth or in detail analysis, so this structure, whatever I have opened, consists of two different chains. So one is chain A and another one is chain B, and length of these two chains are going to be 195. Sequence length means 195 amino acids are there, and the organism is going to be Homo sapiens. Mutations, yes, there is no mutation. Yes, we have confirmed. Now here also, sometimes while we are performing the docking, sometimes what happens is like there are two different chains over here. Sometimes 3, 4 means number of chains will increase. But we need to make sure before starting the study is like whether the both chains are homologous or heterologous. If both chains are homologous, if the sequence is same, in that conditions, we can eliminate one chain. Means assume that there is chain A and B, and if the sequence is same, here you can see the sequence. So if you see here chain A and B, you can notify there is a difference here. See, for example. So if the sequence is same, then we can omit one structure because that will, what to say, that will not consume our time as well as resources. And second thing is like there are likely chances of getting false positive results also. That is why to avoid these things, we will remove the homologous chains and we will keep a single chain. Then we will go for performing docking analysis. So, and this is where the database where we can get the information about this. And these were the few references. Thank you. If you are having any questions, you are you can feel free to interact with me.
Yeah, thank you, sir, for your wonderful session. Dear participants, now the platform is open for any discussion. If you have any questions, you can ask now.
If there are no questions in the sense, either you might be exhausted or it is clear to everyone. Okay, I think there is something in the chat box.
Dear participants, please ask the questions. Sir is waiting. Sir, there is one question.
There is one question in the chat
Yeah,
box by a participant, Monica. So how can I tell how can I tell that the small molecule or ligand docked is an agonist and antagonist? Okay. So one thing I just want to make clear that in terms of in silico techniques, nothing we can say that the docked compound is agonist and antagonist. Here we are going to see only the ligand is having affinity towards the receptor or not. Based upon the interactions, we cannot decide whether it is an agonist or not. But somehow there are few hypotheses or assumptions happening that what they used to do is like they will go for example, if you are going to take cancer only, so assume that for the chosen target, cisplatin is going to be a standard drug. So first cisplatin is going to be docked with the receptor, and wherever the cisplatin has been binded with that particular receptor, that in that particular area, they will generate a pocket. So from that particular pocket, cisplatin will be removed and we will give instruction to our test ligands to dock in that place. So then we are going to compare what sort of interactions our test compound has shown and what sort of score it has shown with that of a standard compound that is nothing but cisplatin. So this can be the approach that can be done. But in terms of a researcher, if you say this molecular docking just give an insights whether it is having an affinity to the receptor or not. As such, we cannot say it is an agonist or antagonist.
Yes, any further questions?
In PDB, sometimes there is discontinuation in sequence. Then what to do? Whether we should select PDB or not. Okay.
Sir, but actually, whatever the ligand we give, it's
Hmm.
showing some sort of binding energy.
Yes, it shows some sort of binding energy.
Then what is the use of molecular docking, sir? Like
So,
I have heard that
yes.
whatever the molecule we give, it is going to show some sort of binding and it is giving some energy. How can we justify?
Yes. So if you see the molecular docking, there are different types of interactions that are being formed between the receptor and ligand. Hydrogen bonding interaction, pi-pi interactions, pi-static interactions means based upon the type of interaction, we can assess the toxicity of that particular compound. This is one thing. The second thing is like, what to say? Score means here we are going to get a doc score, right? Based upon the doc score, we are going to assess whether the particular compound is having a high affinity or low affinity. Based upon the affinity, we are going to assess the activity of that particular compound. This is completely a predictive thing. So that is why mostly when you go for publication also, they will ask you to support the cell line data or animal data to make it very clear. As such, if it is showing some sort of binding, there are most likely getting false positive results. But most of the time this is happening. That is why the journals or researchers, they will ask you to provide some sort of supporting data. For example, same thing has happened in one of our colleagues' research too, where in terms of in silico pharmacology, as you have asked, whatever the compounds we are docking, it will provide some sort of score. But the thinking that it is a good compound, and when we have taken it to this in vitro antibacterial analysis where that compound has failed, it provides a preliminary data, but as such we cannot completely rely on that particular data.
Bharat sir, sorry to interrupt you.
Yes.
Dean sir is waiting to address the participants as well as you.
Sure.
Sir, please.
Good morning, everyone. Yeah, I just want to congratulate all the speakers and all my staff, those who are included in this seminar. And I found that also I received some calls from many of the faculties from the pharmacology. They said the speakers and the topic is very good, as well as it was very important for everyone. So I really want to congratulate again to all the dignitaries, all the speakers, and especially my team, Nagaraju and Moon Buntu sir. They have taken many efforts to get successful this event. So once again, congratulations to all. Thank you very much for participate. All the participants, definitely you all have benefited with this program, and in future also will come with this like this programs also many times. So please give your feedback so we can improve more things, and we'll come with many good topics with this in pharmacology, as well as in coming days in chemistry also, in Sutics also. So thank you very much once again.
Thank you, sir.
Thank you, sir. Thank you so much. Thank you so much for your input, sir. Thank you.
Sir, I think there are few
Bharat sir, you can
questions.
continue. Bharat sir, you can
Yes,
continue.
sir.
Yeah.
Yes,
Thank you.
Monica, one more thing I just want to say. You can just correlate with the lock and key mechanism. Generally, when we are discussing about this drug and receptor action, we will say lock and key theory, right? So just think like only one key is having an ability to unlock that particular lock. But there are several keys that will be similar to that key. It can fit in that particular lock, but they are not having an ability to unlock that particular lock, right? In a same way, several compounds have the affinity to that particular receptor, but only few of them have an affinity to unlock it. So to do that only, we are assessing or comparing with that of a co-crystal, so which is already proven by the earlier scientists. Is it clear, Monica?
Yes, sir.
Question next by Dipali is like PDB, sometimes there is a discontinuation in sequence, then what to do? Okay. So while discussing, we had stated that there are three different methods researchers use for isolating the protein structure. One is X-ray crystallography, another one is NMR, another one is electron microscopy. So in terms of X-ray diffraction studies, assume that this is going to be a product, I mean, this is going to be the three-dimensional structure of a receptor. Wherever the rays are going to be followed, only this particular area is going to be captured in the sequence. The other side of this particular receptor is not being captured, and as such, it is going to be dumped in a database. So in that case, few of the amino acid sequence are going to be missed. So what we need to do is like we need to prepare the protein preparation. We will see before performing the docking, we will go for protein preparation. In the protein preparation, what we will do is like the first thing we will try to fill these gaps, means wherever the amino acids are going to be missed, these amino acids are going to be reconstructed by the use of softwares. Generally, in terms of Schrodinger, there is one option known as protein preparation. In the protein preparation, the missing loops or chains are going to be reconstructed based upon the existing literature. So once this is done, then only we can go for docking. If not, there are chances of getting again false positive results. So once this construction is done, then once again we are going to refine the protein by removing the water molecules which are greater than 5 angstroms, because water molecules are responsible for forming hydrogen bonding interactions. However, if the water molecules are having greater than 5 angstroms, once again, they can show some sort of false interaction. So that is why this is being processed, and then we will go for docking. Is it clear, Dipali?
Yes, sir. Thank you,
Next
sir.
question by Gayatri. If we use free autodoc tools, what are the chances of accepting the results in good impact factor journal? Yes. Yeah, we can. Most of the papers in a good journals that has been accepted by the use of free tools like Autodoc, Veena, and on. There is no compulsion by the journals or else reviewers that only you need to go for licensing softwares. But however, those who are the institutes which are having their infrastructures and all, they will choose licensing because they are more authenticated than the freeware, because their databases are frequently updated. Is it clear, Gayatri?
Yes, sir.
There is no compulsion. You can go with free tools. Most of the papers available in the databases are by the use of free tools only. Okay, next. Vijay, thanks for organizing. Okay. Is there any database which can help to predict targets? TB like SuperPred gives target of human. Is there any database which can help us to predict targets of TB only based upon the structure only? Vijay, can you ask this question? Generally, it is not clear to me. As per my understanding, you mean to ask like, is there any specific database only for tuberculosis targets? This is what you mean to say, or
Yes, sir.
like you mean to ask?
Yes, sir. I am asking that just like we are using binding DB or super pred, right? And that
Yes.
will give us, or that will predict that will give us target, right? Possibilities of protein targets which can be targeted by our compound, right? Based on the smile structures.
Hmm.
So is it possible that we get only targets of TB? Because I tried few databases, right? They are giving us target. Let's say I want to find the targets of any plant chemical, right? But I want to go it for TB, tuberculosis only,
Only
right?
TB you need, right?
I don't want this database. I don't want human targets, right? So, and for infection, that would be true as well. No? For infection, we need to find
Yes,
targets
you
of
need
a
the targets
microorganism.
only for Mycobacterium tuberculosis. This is what you
Yes,
mean to say, right?
yes, sir, yes.
Okay. So as of now, there is only limited set of data available for this bacterial genome sequence. So generally, if see, for example, when I'm saying cancer or Parkinson's or Alzheimer's, the immediately the person used to say there is a database known as gene card where you can get a list of genes that are being involved in that particular diseases. In a same way, for bacterial related gene, there are limited data available as of now. So that is why we cannot state any website which can provide only the targets of Mycobacterium tuberculosis.
Okay, sir. Thank you. Thank you.
Yeah. We can also predict active site, Manjusha. See, we can predict a site where the compound can bind, but we cannot predict whether it is an active site or not. As such, with the help of in silico docking. But this active site prediction can be done based upon the literature, existing literature support. We can select the amino acid sequence, and that we can select and we can say that this is going to be an active site for that particular receptor. Generally, if you see a receptor, there are two different approaches. The one is blind docking, another one is programmed docking. In terms of blind docking, the researchers will not generate any sort of pocket in the receptor. As such, we will dock such that this drug will go and bind in any place. From assume that if A to Z is going to be the sequence of that particular protein, it can randomly bind somewhere. It can bind at A, B, C, D, anywhere. But based upon that, we cannot know whether it is going to be an active site or inactive site. But same in terms of program docking, what we used to do is like, in a first approach, whatever the protein or receptor that we have chosen, initially there will be a presence of co-crystal. That co-crystal we will remove, and around of that co-crystal only we will generate a pocket, and we will instruct the ligands to go and bind in that particular area, and then we will start comparing how good our drug is having an ability to bind compared to that of existing co-crystal. In the same way, even from the existing literature, we can choose the amino acid sequence where active site is there for that particular receptor, and between that particular sequence, assume that if C2 a D is going to be, or C2F is going to be an active site, and Z2E is going to be an inactive site. Means whatever I'm saying, A to Z is a linear structure. But if you see in terms of there, it is going to be a tertiary structure where the protein is going to be in a multiple folded forms. So that is why I'm saying if C to F is going to be an active site and Z to E is going to be an inactive site, based upon the literature, you need to generate the pocket in that particular area, and then you can check whether our drug is binding to that particular area or not. But if you check in that particular case, once again, there are chances of getting false results. The reason is like, even we are instructing to go and bind there. Who knows, the drug can go and bind in another areas also. So that is why most of the time they will go for blind docking also to assess multiple sites where the drug can bind. Is it clear, Manjusha? Muthukumar, how to refine the protein in case of heavy metals are present. Okay. So mostly if heavy metals are present, generally we will go for removal of that heavy metals, because as such, if heavy metals are there, once again, this will interact or this will interfere with the regular interactions that are being formed between the ligand and receptor. But there are few methods where heavy metals are going to be incorporated, but for that cannot be done in terms of freeware. For that you need to have a license where which is a little bit sophisticated one. There you can start optimizing this heavy metals and all. For that also, again, there is a preset of guidelines given. So which sort of heavy metals can be included and which sort of metals not to be. Is it clear, Muthukumar?
Yes, sir, it is clear. Thank you, sir.
Dharani, sir, did my network pharmacology top gene targets come as ATK1 TNF like this, sir? I want to do docking. So how I find my protein of this top gene for the specific disease? Dhanashree, can you explain? Generally, this question is not clear to me. Can you explain? Top gene target comes as ATK1, TNF, and SRT. Okay, you have performed network pharmacology, and based upon this, you have identified these were your targets. Dhanushree, can you turn on your mic?
Yes, sir.
Yes. So
Sir, I did my network pharmacology.
okay.
This ATK1, TNF alpha, SRC, like this top target are come. So
Okay, you got several targets. Okay.
yes, sir. So I want to do docking. So how I find my targets? How I find proteins of this target?
See, first already you have stated in network pharmacology, you have got few targets, right? So that targets you need to first rearrange as per their score. So please be more specific. You want to dock, means you want to check all the proteins that were involved in that networking, or you need to categorize based upon their ranking. Generally, they will go for top 5 to 10 targets that they got. So that target names, whatever now you are saying, like here ATK, A1 and TNF alpha, this once again, these names you need to type in the database as I have shown how to search for MMP9. In the same way, you need to check for your particular interest of targets in the PDB. So there, based upon the criteria that we have discussed. So one is resolution, another one is species, another one is existing of a crystal. So based upon these criteria, you need to select your PDB. Then for this particular PDBs, whatever the ligands that you are planning to dock, both together you need to combine and you need to perform docking. This is how we used to do.
Okay, thank you, sir.
So, hello, sir. Can you suggest some good impact factor journals for network pharmacology related research papers? Okay. By Anjali. As such, if you see for network pharmacology alone, there are really limited papers. Journal of Molecular Modeling, Journal of Structural Bioinformatics. There are like very limited journals, because nowadays what happened is like there is shift in the research papers. In the era of COVID, every one of us mostly they have focused on this in silico data, and it became a huge in silico data available in their publications. Now, when they are going for replicating the same in terms of cell lines, the results are not exactly matching. So that is why nowadays what all these journals are asking is like, at least they are asking whatever the data that you have done, at least they are asking for one type of supporting data that might be either in terms of cell lines or in terms of animals. As such, the acceptance rate for in silico modeling or network pharmacology is very limited. But there are few journals, as I stated, Journal of Molecular Modeling, Journal of Biomolecular and Structural Dynamics. Then we are having RSC Advances also, few of the papers got accepted, even ACS Omega. But it depends upon the hypothesis, how good you are going to frame once again. Without any supporting data also they are accepting, but it depends upon the hypothesis, how strong you are going to frame. Is it clear? Okay. Next, Pradeep, what points to be considered for writing discussion of network pharmacology research paper? Can you please elaborate molecular dynamics? What are softwares used in free? Okay. So in terms of network pharmacology discussion, what need to be written in a research paper? So this is a regular thing, what and all the initially we had seen a different types of or different stages of network pharmacology, right? So all these things you are going to state over there. But apart from that, what makes your paper more identical is like, you need to be more specific towards the lacunas what your paper is having. So the first thing is like how you have framed the hypothesis, that goes in an introduction section. So in each of the result and discussion section, you need to correlate it with the existing literature, and you need to state what sort of lacunas that you are observing, which means you are not biasing the data what you have got. You are mutually stating what benefits you got with the study and what sort of lacunas also associated with the study, which will enhance the quality of your paper and trust on your paper, such that even the other researchers who are going in the same part cannot like they can't waste their time. So this is how you can enhance your quality in terms of what is it, results and discussion. The next thing is like, once the molecular docking is done, further they will go for molecular dynamic simulation studies, and you are asking about the three tools that are there. So the first thing, in terms of molecular docking, when we say, I have stated that ligand we are taking and receptor we are taking, we are going to dock together and we are seeing the affinity. So in molecular docking, both the ligand as well as the receptor, both are in a rigid state. But in terms of body, if you see, once if a drug has been taken or any endogenous compounds goes and bind to a particular receptor, there will be some sort of conformational changes happening. As such, it won't go and bind in this way. They will start means they will start rotating, and they will go in a flexible manner, and they will start binding, such that this complex will start undergoing a conformational change, right? Again, in the body, there will be existence of several ions, water molecules. So how long this particular interaction is going to be stable? So whether there are any other ions that can influence this binding capacity or not, that we are going to assess with the help of molecular dynamic simulation studies. Generally, for molecular dynamic simulation studies, we will go with Desmond, Jaguar, sorry, this is under Schrodinger, Jaguar. But in terms of freewares, most commonly used freeware for molecular dynamics is GROMACS. GROMACS is the most commonly used freeware. It is completely free, but for that you need to know few lines of coding, which is like you can freely get it from either YouTube, or a few documentations are all available. So if you follow those particular guidelines, you can easily complete your free dynamics. But however, for that you need a good computational facility.
Yes, I think all the questions,
Yes, sir.
all the questions are addressed.
Sir.
Audience from the participant side, can anyone or few please unmute yourself and give the feedback about the overall workshop, and what are the improvements we need to have? Do you require any hands-on workshop, and what are the things you want to have in the workshop? If you can give those inputs, we can design a better workshop in future. Please.
Hello, sir.
Yes, sir. Go ahead.
Yes, sir.
Yes, sir, it's me, Aman Khandelwal, research scholar from JSS College of Pharmacy, Ooty. Although I have some knowledge of network pharmacology in advance, but this workshop has vastly helped me in gaining more knowledge, and it has refined me in this field. And I had so many doubts which all got cleared, and thanks to all the speakers also, and thanks to Sandeep University for providing such opportunity. And if possible, please conduct such more workshop in future, and if possible, please conduct a hands-on training also for network pharmacology, so it will be helpful for us in future, sir.
Sure, sir. We'll take into that. Any other participants?
Hello. Uh, yes,
Yes,
am I
sir. Go
audible?
ahead.
Okay, great. Thank you so much to the Sandeep University and the speakers as well. All the speakers were renowned and they had groundbreaking knowledge in the field of network pharmacology. So, of course, thanks to the university that they picked up such speakers, such renowned and very well knowledge containing speakers, and they gave us lots of knowledge about network pharmacology. It was too much, we can say, filled with knowledge. Okay, so thank you to the team and also the speakers.
Thank you, sir. Thank you, Vijay, sir. Any other participants?
Hello, am I audible? This is Dr. Shiva
Yes, sir.
from
Yes,
Gujarat.
sir. Yes.
Sir, in a nutshell, to make long story short, this workshop take into another venture of what you can say the network pharmacology, because most probably I may feel like the participants are already into the docking and simulations or connected to the in silico in some or other mean. But this workshop is a kind of fun. I don't know about others. Like I have attended for the first time. They'll be revealing end to end, starting from the source ending up to the freewares of servers, free web servers that we can able to adapt for the purpose of ADME or Swiss ADAC, or with respect to protein identification, or with respect to target simulations and everything. And it's really a brainstorming. Thanks for your university and also the organizing committee, very kind and polite enough. And also at this platform we should appreciate the audience who maintain the discipline of the quorum to the maximum without causing much of interruptions. They allowed the presenters to expel the knowledge of the maximum to its capacity. And thanks to the organizing committee, and also thanks to all the resource persons from day one until now. They are actually very kind enough in taking detailing of each and individual question has been asked by the participants at the end of the show. And they also shared their email ID for future communication to get happen. It is not only a network pharmacology by its kind, it also a kind of faculty network most probably will happen through this forum. Thank you once again.
Thank you, Dr. Shivaram, for your valuable inputs. Truly, the best thing about the workshop is the participants. They have maintained the decorum. They have been patient. And the first day we have got few technical glitches. Even then, you are very cooperative. Thanks for that. Any other participant? Any other participant? Because it is really a driving force for us to conduct more similar workshops in future. Yeah, if there are no one to give your feedback, we will have the last part of the workshop. I would like to invite Dr. Ramdas Dola sir, Associate Dean, School of Pharmaceutical Sciences, Sandeep University, to give the vote of thanks.
Good afternoon, all the participants. As we come to the end of this 1 week national e-training program, it is always said that well begun is half done. So I take this opportunity for giving the oath of thanks for the 1 week national e-training program on integrating network pharmacology with traditional and modern systems of medicine, bridging multi-system therapeutics for holistic healthcare. I take this opportunity to express my heartfelt gratitude to everyone who has made this event a resounding success. First and foremost, I extend my sincere thanks to our chief patron, Dr. Sandeep Jha, Honorable Chairman of Sandeep University, for his constant support and encouragement. Your vision and leadership has been instrumental in forecasting as well as fostering such knowledge sharing platforms. I am profoundly grateful to our patron, Professor Dr. Rajendra Sinha, Vice Chancellor, for his guidance and for setting the tone for this program. I would like to profusely thank our OSD, Professor Pramod Karole, for his insight and guidance. I would also like to thank Mr. Vivek Nikam sir for his support. And a very, very big thanks to our esteemed resource persons, Dr. Anup Kumar, Dr. A. Shekhar Reddy, Dr. Diwakar Shelvaraj, Dr. VVS Prasanna Kumari Rayala, Dr. Akai Krishna Sarup, and the today's speaker, Dr. S. Bharat Kumar Reddy. Thank you all for sharing your expertise and illuminating various dimensions of network pharmacology. Your sessions have provided valuable insight into bridging traditional and the modern therapeutic approaches. And even the feedback which was given by the various participants, it was really, and we can say, a boosting for all the university people of Sandeep to arrange such a kind of programs further. A special thank to the organizing committee under the leadership of our dynamic dean, Dr. Makaranda S. Gambire, Dean and Professor, School of Pharmaceutical Sciences, for his seamless executing programs. I acknowledge a special thanks to the organizing secretary, Dr. Mohan Gandhi and Dr. Nagaraju Bandaru. So I think that both of them, they need a round of applause from our side. So I request all the participants that we should clap and we should motivate both the organizing secretary that they should carry on with such a kind of training programs in future also. So both Dr. Mohan Gandhi as well as Dr. Nagaraju Bandaru, they have given their efforts in ensuring the smooth coordination of this training program. At the last, I express my thanks to all the participants, students, faculty members, researchers, and industry professionals who have actively given their engagement from their soul for making this event successful. Thank you for your enthusiasm as well as throughout contribution. Lastly, I extend my gratitude to the technical team and the support staff whose behind the screen efforts and ensure the event run flawlessly. Let us take forward the learnings from this program and continue our journey in advancing multi-system therapeutics for holistic healthcare. I thank you everyone and I wish you all have a wonderful day ahead. And I request all the participants to please stay in the meet because there are some instructions which the organizing secretary would like to give you people. I hope you all have enjoyed this 1 week training program, which has been organized by the Sandeep University Nashik. And this may have helped you in gaining the various insight of the pharmacology. And when we talk about the various aspects, I was just listening to the feedbacks which were given by the various participants. Really, it is a motivating factor to all the team members of the Sandeep University to arrange such a kind of programs further. So on behalf of Sandeep University School of Pharmaceutical Sciences, I assure you that definitely if you people are willing to attend such a kind of program, we are there to arrange such a kind of program with you. Now I hand over this to the organizing secretary, Dr. Mohan Gandhi. Gandhi sir, please.
Uh, Apologies from my side. One more program is left.
Sir,
Yes.
can I quit or?
No, sir, sir. Please wait. Please wait. Please wait.
Yes,
Yeah,
yes,
sir,
yes.
sir,
Okay.
please.
Okay.
So, Bharat sir, thank you so much for your presentation. As a token of appreciation, please accept.
Thank you very much, sir. On this platform, I just want to extend my gratitude and thanks to Dr. Ake Krishna Swaroop and Sandeep University for providing a platform for me to be here today. And I also extend my special thanks to Dr. Vidya Srileka and Mr. Prudvi Penumula for helping me out to create such a wonderful presentation for today's session. Thank you very much, sir.
Sir, from personal side, I would like to ask you one question or one request.
Sir.
Sir, is it possible for all the participants or from Sandeep University that can we have a collaboration with you? Like if any participant has any query regarding their research or regarding the publication, whatever it may be, because being very close to you, I have taken the liberty in asking this question on the open platform.
Sir, as we have asked on an open platform and it is good for me and I'm saying it very clearly, I'm always happy to collaborate with any one of you. So because it is somehow helping me to brush up my existing knowledge and somehow it also helped me to learn new things. I'm always happy to collaborate with any one of you from this platform, sir. And I hope my mail ID is there with you. And few of the participants were also asking, you can share my mail ID too. I'm always happy to help the things which I know. And I'm
Thank you,
also
sir. Thank you.
happy to collaborate.
Thank you, sir. Thank you so much.
Thank you, sir.
Participants, that's it from our side. The workshop, I personally feel it is very informative and very fruitful. And all the resource persons are very cooperative and very, humble that they have accepted our invitation and they have even accepted our request to collaborate with all of us. So if any participant from any college, irrespective of your specialization, if you have any queries or anything in your mind, you can directly contact them through email and they would definitely respond to you. And over to you, Nagaraj sir.
Sir, and sorry to interrupt. Even this also
Yes,
I'm making it clear on this open platform as the research, my research is not only confined to this particular in silico pharmacology and network pharmacology. So I'm having a diverse set of expertise. Not only me, I think even all the resource persons, I think they might be having a diverse set of expertise. And one more suggestion even to these participants is like, don't confine to one particular field because exploring your research in a diverse set will mostly help you to procure some sort of funding. The other thing is like when you are planning to go for any sort of higher studies that might be either PhD or postdoc also, a diverse exposure will help you to get familiarized with different other aspects of other departments too. So it's just a request.
Sure.
Yes, sir. Thank you, sir. Thank you, Bharat Kumar Reddy, sir, for your beautiful and knowledgeable session. So most of from the feedback of the participants, so we will conduct the offline training session or maybe workshop for this in the month of maybe February or March, definitely. Okay, so hope you please stay in that WhatsApp group only. We will communicate in that.
Sure, sir. Sure, sir.
Yes.
Sure, sir.
Thank you for all my dear participants. So your continuous support and encouragement. Thank you. Thank you. And we will send the certificates in the next.
Thank you.
Yes.
Thank you, sir.
Sure. Thank you. Thank you all the participants. Thank you, Bharat sir. Thank you so much.
Thank you, sir.